Hello, my name is Brittany Parker, and I will be presenting on Huntington's disease mechanisms. Unfortunately, I am trying to videotape this presentation from a hospital room right now, but just like those who have been affected by this disease, I'm just going to deal with the cards I've been given and push on through. So, what is Huntington's disease? Huntington's disease is a rare neurodegenerative disorder that affects a very small portion of the population. It's caused by an accumulation of a protein called Huntington. The accumulation of the protein Huntington affects mainly one part of the brain, but the part of the brain it affects controls motor movements, emotions, and cognition. So who is affected by Huntington's disease? As I said earlier, it's a very small population. 1 in every 10,000 people in most Western countries is affected, and 3 to 7 per 100,000 people of European descent are infected. That doesn't necessarily mean that every other ethnicity, race, origin doesn't is not affected by this disease, but it's so small that it's almost negated. It mostly affects people of European descent. The onset of disease usually happens between the ages of 30 and 50, but there's a very small percentage, 6% of individuals who have onset of the symptoms and the disease very early in life, children and teenagers. So symptoms of the disease. So there are basically 3 major areas that the disease affects. So movement would be first. So things such as walking, the speed of your gait, speech, swallowing. There's a symptom called dystonia, which is an issue with the contraction of your muscles, so you get a little rigid. There's also chorea, which is muscle jerking and the writhing motion that you sometimes see in Huntington's patients. As far as cognition, there's difficulty with speaking, difficulty with comprehending, difficulty with multitasking, impulse learning, new behaviors, and awareness of their own behaviors. Next would be the emotional. There's depression, social withdrawal, sometimes a complete 180 in the person's personality. Someone who's usually docile may become aggressive and irritable. Insomnia, fatigue, sometimes manic fits, and even obsessive compulsive disorders. So to the meat of the presentation, which is the mechanisms of this disease. So Huntington, the protein, as I stated earlier, is what actually causes the disease. The exact function of the protein is still actually unknown, but it appears is very vital for neuron function. It may be used for cell transport and protection against apoptosis, which is cell suicide, but again, it's not 100% sure. While in its normal size, the protein functions normally, everything in the body functions normally, but when you have the disease, the protein has an altered form which causes the disease. And it causes the protein to aggregate, which means to clump up, and once it clumps, it becomes a toxin. So, where does disease come from? It comes from the HTT gene. The HTT gene encodes the protein Huntington, as I stated earlier. It's located on the 4th chromosome. The mutation in the gene causes an overproduction of the CAG trinucleotide. So this disease is a trinucleotide repeated disorder. Normal genes have about 10 to 35 CAG repeats on the 4th chromosome. An abnormal one will have between 36 and 39, and individuals with this abnormal gene may or may not exhibit some signs of Huntington's, but definitely individuals with 40 to maybe even 120 CAG repeats will definitely have Huntington's disease. And as you can see in the photo, this is where the gene is located. So, the CAG repeats cause a abnormally long glutamine tail, which is what CAG codes for. This long, sticky tail causes the neurons to become rigid, which is not normal. They're usually very fluid, and it also may block other proteins from from functioning properly. Again, there's research being done on this, but there's not 100% sure of exactly what the protein does or what the toxin does itself. So this is what its effects on the brain. This picture demonstrates the basal ganglia. The disease particularly affects the basal ganglia. And when you find out what the basal ganglia actually does, it makes sense why the disease has the symptoms it does. The basal ganglia controls motor function, capacity for procedural learning, emotions, and cognition. Atrophy in this part of the brain is what causes the symptoms of this disease. So on to diagnosis and treatment. Diagnosis of the disease can be in several different ways. There's always the neurological exam to test for symptoms, which is basically just going to the doctor, and once you have the symptoms, which is depression and maybe some slurred speech, clumsiness, again, maybe a 180 of the personality you had before, and then from that point, the doctor would either do brain imaging or even just directly test your genes and find out if you have the mutation in your HTT gene. Also, family history plays a major part in it. If your parents had the HTT gene, or if they did in fact have Huntington's disease, then more than likely you have it as well, because anyone who has the Huntington's disease has a 50% chance of passing it on to their children. So for treatment of Huntington's disease, there is no cure, and there's actually no treatment for the actual disease. There's nothing to slow the progression of it or the atrophy in your brain. All they can do is simply treat the symptoms. So physical and emotional therapy are the most basic of the treatments. So physical therapy for the muscle rigidness and the chorea. Emotional therapy, of course, for the depression. Tetrabenazine is one of the major drugs used in treatment. It treats the chorea, and it treats it by inhibiting monoimmune uptake from the neurosynaptic system. So essentially, as I said earlier, the protein is aggregating, and that's why you have the symptoms that you have. So when you have a monoamine uptake and you're having it suppressed, you're helping to stop the proteins accumulate as much. Although you're stopping the proteins accumulating as much, it still is not slowing the progression of the disease, however. Antidepressants are used, of course, for the depression. Antidepressants such as sterline and citalopram are used. Antipsychotics such as risperidone and quetiapine are used, and those are used for some other symptoms that come along with the emotional problems, which could be manic fits, obsessive compulsive disorders. And then you have mood stabilizers also for the emotional problems, such as valproate and lamotrigine. So what research are we continuing to do to find out more about these mechanisms and how we can treat the disease? The most leading research is in animal models. Most diseases that are neurological, it's hard to test because the human brain, although not extraordinarily unique, it is unique, and diseases that affect our brain don't necessarily affect other brains the same. Other animals' brains the same, excuse me. Mice, for example, is a really good model to use because they have a smaller chromosomal database, so you can measure and experiment on a smaller scale. And there's also, like I said, so many unanswered questions. We still don't know what the exact function of the protein is, why the altered form of this protein is so damaging, and why are the symptoms onset so late. Another very interesting thing I saw in research was that there is leading research right now in treatment with medical marijuana. And it's, again, another treatment that doesn't necessarily help to slow the progression of the disease, but more so works with helping to aid in the entire process with emotional problems, depression, and everything like that. And also, the medical marijuana has been proven to help with chorea without the bad side effects of tetrabenazine, which can sometimes be to aid in the depression or even to have an exact opposite effect in some patients and not work at all. These are the references I had. Thank you very much for listening to my presentation. Again, I apologize for the quality and where I have to be, but again, you deal with the circumstances you get. Thank you.